So, the title of our research is investigating the therapeutic potential of blocking IL-1R1 cytokine activation in chronic inflammation-associated prostate cancer. The presenters for this research include Charlanda Metayer, Ariana Perez, Corey Nar, and Preetika Praveen. Our mentors include Dr. Sahito Seni and Dr. James Kumidiaka. I will first begin with the purpose for our research. For decades, interleukin-1 family molecules, cytokines and receptors have been known to be important molecular drivers of inflammation and immunity. Both interleukin-1 alpha and interleukin-1 beta bind to the interleukin-1 receptor type 1 and initiate downstream pro-inflammatory signals via activation of NF-κB transcription factors. Prior studies have shown that the upregulation of IL-1R1 contributes to the tumor initiation and progression in multiple myeloma, head and neck squamous cell carcinoma, testicular, pancreatic, breast, and colorectal cancers. The repression or blockade of interleukin-1 R1 activation has been clinically confirmed to inhibit tumor growth and angiogenesis in patients with these cancers. Whether such effects could be replicated in prostate cancer is unknown and yet to be explored. The therapeutic potential of anakinra, the most commonly used recombinant IL-1R1 inhibitor, in combination with taxane analogs, conventional chemotherapy for advanced prostate cancer, for the treatment of prostate cancer, has not been explored as well. Hence, in this study, we aim to investigate the effects of blocking IL-1R1 activation with anakinra on prostate cancer cell growth and metastasis, as well as the ability of anakinra to potentiate the anti-cancer efficacy of taxane analogs. We hypothesize that downregulation of or blocking the activation of IL-1R1 will inhibit prostate cancer cell proliferation and metastasis, as well as induce cell death via apoptosis in a dose and time-dependent manner. My colleague Preetika will now present the background.
Prostate cancer remains the most diagnosed cancer and the second leading cause of death in U.S. men. In 2020, an estimated 191,930 new cases and 33,000 deaths from prostate cancer are expected among men in the United States. Though a lot of therapies have been developed to manage the early stages of prostate cancer, including androgen deprivation therapy and radiation, most prostate cancer patients often develop biochemical relapse or resistance to chemotherapy and radiotherapy, which leads to advanced or metastatic progression of the disease. Chronic inflammation has been suggested as one of the possible molecular drivers of prostate cancer progression activation of multiple inflammatory cascades or pathways, including those involving the interleukin-1 family molecules. The interleukin-1 family is a group of 22 cytokines and receptors that mediates the regulation of immune and inflammatory responses, acting as either pro-inflammatory or anti-inflammatory molecules. These inflammatory responses may play influential roles in inducing tumor initiation, promotion, malignant conversion, invasion, and metastasis.
I am Charlanda Metayer, and I'll be presenting the methodology. Interleukins are associated with chronic inflammation, so we did a bioinformatics analysis to look at gene expression in clinical prostate cancer patients and found that interleukin-1 receptor type 1 is highly expressed in these patients. For our experiment, we are doing 2 in vitro studies with different prostate cancer cell lines to see how inhibiting interleukin-1 receptor type 1 affects the growth and metastasis. We are using 2 cell lines, LANCAP, short for lymph node carcinoma of the prostate, and PC3, short for human prostate cancer 3. A Western blot will be performed for each cell line to measure the expression of interleukin-1 receptor type 1. They will be cultured in media containing 10% FBS and 90% RPMI 1640 medium. Then we will put the cells in tissue culture flasks and incubate them at 37 degrees Celsius and at a carbon dioxide level of 5%. In a 96 well plate, we will seed 10,000 cells in each well. Anakinra is the drug we are using that will serve as the interleukin-1 receptor type 1 antagonist. We will treat the cells with different concentrations ranging from 1 microgram per milliliter to 10 micrograms per milliliter for 48 hours. Then the MTT assay will gauge the effects of our treatment on the cell's viability and proliferation. Absorbance will be used to find the concentration of anakinra that kills 50% of the cells for each cell line. The colony forming assay will determine the survival rate of these cell lines after 7 days. We will measure the metastatic potential and cell death with scratch migration assay and acridine orange ethylene bromide dual fluorescent microscopy. We will do another Western blot to see how interleukin-1 receptor type 1 is expressed in the cells after they've been treated with anakinra.
For our statistical analysis, All results will be generated in triplicates and expressed as means plus or minus standard deviation. The significance of differences will be measured by student t-test and one-way ANOVA to test our hypothesis and also show the differences of components between the control group and the drug-treated group. A p-value of less than 0.05 will be considered statistically significant. Each experiment will be repeated independently for at least 3 times.
Hello, my name is Ariana Perez, and I will be presenting the anticipated outcome. Since interleukin-1 family cytokines have been suggested as one of the possible drivers of chronic inflammation-associated tumorigenesis, we carried out a transcriptomic bioinformatics analysis on RNA sequence datasets of prostate cancer patients acquired from TCGA and C-Bioportal databases. In that study, we profiled the expression pattern of 22 interleukin-1 family genes in 493 patients with local and 230 patients with metastatic prostate tumors, respectively. In this image, Figure A shows the high expression of interleukin-1R1 in the early stage versus in Figure B, the high expression of interleukin-1R1 as well. Our preliminary results indicate that 9 out of the 22 interleukin-1 genes are highly expressed in both groups of prostate cancer patients, and their overexpression may be contributing to the progression or relapse of the disease. Interleukin-1R1 was found to be the most overexpressed interleukin-1 receptor in the prostate tumors of these patients compared to adjacent tissues as visualized by the heat map generated using the ggplot2 package in RStudio. We also analyzed interleukin-1R1 immunohistochemical staining from prostate cancer tissues deposited in the Human Protein Atlas database and found interleukin-1R1 protein expression to be highly expressed in high-grade prostate cancer patients. In Figure A, we see the high expression of interleukin-1 protein, which is in the brown color, in prostate tumor cells of high-grade prostate cancer patients. In Figure B, this is a low expression of interleukin-1R1 protein in prostate cancer tumor cells of low-grade prostate cancer patients. In Figure C, we see the normal type where no brown color is expressed. Based on our preliminary data, we anticipate that blocking interleukin-1R1 expression in prostate cancer cells with anakinra will inhibit prostate cancer proliferation, viability, metastatic potential, and colony formation ability, while at the same time inducing apoptotic cell death in a dose and time-dependent manner. We expect that treating tumor cells with anakinra may enhance or potentiate the therapeutic efficacy of taxane analogs, thus acting as a adjuvant therapy.
We would like to give a special thank you to Florida Atlantic University and to our mentor, Dr. Sahid Oseni, and our advisor, Dr. James Kumidiaka. We thank you kindly for listening.